Hello, my name is Christopher Bravery. I'm a consulting regulatory scientist at Advbiols. And today I'm going to talk to you about excipients. So I'm going to talk about selecting excipients, formulation studies, novel excipients. Before I start, I just want to comment a little bit about terminology. Many of you probably heard me say this before, but GMP isn't a grade, it's a quality system. So you shouldn't use that term because it's actually meaningless. To my knowledge, there's not per se any legal requirement for GMP, but we do need to remember that excipients, unlike raw materials, there's a deliberate intention and to administer them to the patient. So they do have to have a slightly better quality, potentially, than raw materials. So be aware of all the sort of terms that are often used by suppliers, like chemically defined, animal component free, etc. Remember, there's no harmonized meaning for these, and indeed, they're sometimes not quite what they seem to be. They sometimes gloss over details that you really want to know. So I'm going to start the body of my talk by giving you my general regulatory principles for excipients. So these won't be written like this, it's so succinctly in any regulatory guideline, but I think this really encapsulates the way you should think about excipients. So each excipient, and by which I mean the individual substance, not the whole formulation, each individual substance should have a purpose. So examples of purposes, and the list here isn't much longer than this if I included everything, would be tonicity, stability, pH, antioxidant, cryoprotectant, that sort of thing. If you got tablets, then there would be other things in there as well. But we don't do tablets, these are parenteral. So the corroboree of that is if it has no purpose, it should not be there. So the second principle is that each excipient should be present in the smallest quantities required to have their effect, because they're deliberately administered to humans. And this is even if they are innocuous, harmless, completely safe, that doesn't matter. They should still only be there in the minimum quantities. Excipients also need to be compatible both with the active substance, particularly when you're thinking about things like cells, viable cells, the container closure system, and obviously any delivery device. And actually what I just suddenly realized I missed off here is obviously biocompatible with humans as well. Hopefully that's a given. But actually things like, you know, the wrong pH and things can affect things like discomfort and pain and reaction site events and so on. And when you justify the use of the excipients, if you basically justify them applying these sort of principles, then you'll cover everything that a regulator wants to know. So there isn't a huge amount of guidance in this area, and the main guide really is ICH Q8. I've given you the FDA version where they just call it Q8. ICH Q8 talks about formulation development, and I found this table in the training materials, and I think it was a sort of useful one to show. I'm not going to walk through the entire thing, I don't have that much time, but I think a key thing is that it's trying to put Q8 in the context of an entire quality risk management type approach, and a risk-based approach to excipients is obviously the right approach to take. So hopefully you would have a target product profile for your company, for your product, I mean, your company should have one. And from that, you would develop a quality target product profile, ideally. It's not essential, but it's highly recommended. And that would include aspects that would be relevant to your decisions about what you require your formulation to achieve. So to take a very simple example, from a commercial perspective, it might be essential that your product has a minimum, say, 2-year shelf life. And so that would be in your quality target product profile and would give you an objective of your formulation development. You may have a preference that the shelf life would be 3 years, and therefore you might work towards that, but as long as you got 2 years, you would feel you'd done your job, sort of thing. It talks about a few different stages here. Pre-formulation studies, these would be done in preclinical development. Remembering that preclinical toxicity studies should really be done with the same formulation that will be taken into man. That might not be the final formulation at approval. And indeed, what I'm describing might be iterative over development. I have for biotech seen liquid formulation followed by a lyophilized formulation, followed by a pre-filled syringe. It's liquid again. However, we don't tend to do that too much, and I imagine that's quite a lot of work, so I'm not suggesting that's the right thing to do. Then formulation screening, I'll come on to that because that's really formulation development studies, if you like, and then optimization and so on. So that is quite useful. So let's quickly move on to quality. Now, all the same principles that apply to raw materials apply to excipients, and then you have to add some more considerations on. So I'm not going to run back over what you would normally talk about with raw materials. Those are just a lot at conferences. So hopefully you feel comfortable with that. Of course, light raw materials, it's helpful if you can use pharmacopoeia grade materials because that really saves a lot of effort in the dossier. For example, in P4, which is where you describe your excipients, which is analogous to S23, where you describe raw materials, you wouldn't have to actually provide a specification if it's a pharmacopoeia grade because it's in the pharmacopoeia, so you just state it's pharmacopoeia grade. Otherwise, you have to list out the entire specification. And if it's an in-house, that means you're also having to provide all the analytical descriptions of all the analytical methods that you use to release that material, and all the validation of those methods as well. So using compendial methods is also very helpful here because, again, you don't have to do that description and you don't have to do the validation. The next slide is really here just to remind you of the various categories of quality attribute, if you like, that you should consider for any material, just to sort of remind you to make sure that any specifications for non-compendial materials are complete. So now let's move on to actually justifying the specifications. So the first point I've already made, which is you need to justify the choice of the individual substance, the individual excipient, based on its purpose. So that should be there in your justification. So that would also be with a raw material, but obviously slightly different. Then you need to justify the quality of the excipient in a similar way you would do for raw materials. So that considers things like purity, risk of impurities and adventitious agents, etc. Details of adventitious agent safety in A2, just like raw materials. And then you need to consider the risk to the patient as an excipient. So it should be inactive in the concentration used, but it may have some other sort of toxicities, and they may be minor, they may be major, and it may not be that there's an alternative. So DMSO being an example, really, that we don't really have a good alternative, and there are toxicities associated with it. Therefore, your formulation should consider the limits of that toxicity, i.e. using the minimum amount required, for example. You may get some data from preclinical toxicity studies where you've administered the product in animals to look at toxicity. If you have a novel excipient, I'll touch on this again before the end of the talk, then you may need to do specific toxicity studies for that. And then there's the literature that will help you justify the safety of the excipients you're using. So I have a copy of this book. It's a little bit old now, but actually I find it really useful, not just for excipients, but sometimes for other materials as well. One of the things I like about it is it explains where the material comes from, how it's manufactured and prepared. So DMSO, which I've chosen here as an example, you may or may not know that that's a byproduct of paper manufacture, for example. It gives you the the compendial specifications, both for Furrier and USB. You can see from this that it is old because that's Furrier 6 and we're up to 10 or something now. What's quite useful about putting them side by side for you is it helps you see where there are differences between compendia. I mean, they try and work on compendial harmonization, but it's an ongoing program. It gives you some chemical details, and then often it will give you this sort of useful information about known toxicity of the substance, and it will give you references and so on. So you will be able to use that in your own justifications. Then another source of information, which is only relevant to the FDA in that I'm not sure whether this could be cited to the EMA. I suppose you could, but it's not really going to be necessarily have the same impact. But for every product that the FDA has approved, they have included in this database the excipients used and some information. So if you can find the excipient that you're using and the route of administration you're using in this database, then I'll show you an example, then you'll be able to conclude that it's not novel. It doesn't really give you a lot of other information. In theory, there's a column for maximum daily exposure and another one I can't quite remember, and it's off view. But I found that there's very rarely ever anything in those columns. But for the examples that are there, it usually gives you the maximum amount of this excipient that the product that it's used in exposes the patient to, if that makes sense. I hope I explain that clearly. And here's poloxamer 188 as another example. So quality systems. So we dealt with the quality of the excipients themselves, albeit very briefly. What about the quality system? Well, I'd like to draw your attention to a very nice technical report from the Parental Drug Association, and its title is Formalized Risk Assessment for Excipients. And that's what it does. It goes through in quite a few pages and in a lot of detail how to go through a whole risk assessment, not just from the regulatory point of view, but also commercial, so things like supplier audits and all that sort of stuff. And what they have suggested in this document is a very pragmatic view to the quality system of the excipient supplier, or manufacturer preferably, I suppose. Where there's little risk mitigation required, i.e. my example that I've added onto here is perhaps sodium chloride, where we know there's no toxicity risk. Well, actually, there is a toxicity risk with sodium chloride. If you've got kidney failure, then your doctor, and you were going to infuse a large volume of something with sodium chloride in it, then they would have to consider the impact on your kidneys. So don't completely rule out even apparently innocuous things like sodium chloride. You have to think about it in the context of your product. And then a higher risk material might be something like recombinant albumin. Where, of course, there's adventitious agent risks, et cetera, and it's manufactured from a mammalian cell line and all that. So in that situation, clearly you would want GMP. It's worth also touching on, of course, that the FDA allow the use of a type 4 drug master file for excipients. I just refer you back to my principles and think about how well that complies with my principles. There isn't an equivalent in the EU, and actually they will expect to see all the details in the dossier. So you may have to work with the supplier to make sure you can get adequate details. Then moving on to formulation development. Now, the thing about formulation development is I see it very rarely done for cell therapy products to date, in my experience. I'm more likely to have seen it with a gene therapy vector. And indeed, when you look at the published literature, there's a fair bit on viral vectors, and I didn't easily find any formulation studies for cell therapy, actually. I'm sure there's some out there, but preparing this, I didn't immediately find any. So the usual approach for other biological products, every other one I've ever worked on other than an ATMP, is that you do a load of formulation studies and figure out the best formulation for your product. Because an off-the-shelf formulation is literally that, is the difference between having a suit tailor-made and a suit off the peg. It might fit, but it might be a bit baggy in places. And the point is that even two monoclonal antibodies or whatever will have some differences, which might require some little tweaks to the formulation. So just taking something off the shelf really isn't ideal. And then you learn a lot from doing these formulation development studies. Now, one of the reasons I raise this is only because just recently on a couple of occasions, I've read in-house formulation development referred to as homebrew. And to me, it was inferring that it's somehow inferior, whereas my argument is it's a superior way of doing it, because then it's tailored and optimized to your product, your route of administration, and possibly even your indication. But this is not to say that you couldn't work with a supplier to optimize an existing formulation. Indeed, I do know that some companies have done that. So what's expected? Well, you're following the regulatory principles that I've described, and you should be exploring possible different excipients, different concentrations, etc. You're trying to find a combination of excipients that gives you good stability, good biocompatibility, is suitable to administer to humans, etc. So these are not necessarily easy studies, and they're likely to be aided by things like using design of experiments and things to minimize the number of replicates and so on you have to do. Commercially, you may have some other considerations like costs and supply risk, for example, as well. So nearly at the end, and just two quick slides of simple examples. They both AAV, but they're from different papers. And it's just to show you the point of the sort of studies that you would do for formulation development. So here on this figure, we've got the average particle radius. So it gives you an idea of aggregation, etc., I assume. Osmolarity and ionic strength, and then some different sorts of salts, buffer solutions, etc., looking for what gives them the best characteristics. And likewise, this second example, which is a very fuzzy image, and so a little bit difficult to see the symbols, but it doesn't really matter. I've added actually the formula of poloxamer 188, just so you can see what it is. But those of you working on AAVs will probably be familiar with it. It's very good at stopping aggregation. And as you can see, they're looking at a combination of different devices and formulations that do or don't include pluronic F68. And the first few symbols up the top are around about 100% of the normal concentration, but the little cluster to the right there have all dropped well below 100% and clearly are not suitable formulations, so they wouldn't be taken forward. So brief, I'm sorry, but hopefully that provides you a good overview. And just to summarize, excipients need to be need more careful consideration than raw materials because they're going to be administered to the patient. You need to think about toxicity to the patient as well. Off-the-shelf formulations might seem simpler, but they will not necessarily be optimal for your product because every product is unique. But this also limits knowledge that you will gain otherwise from doing formulation studies. And it might make it difficult to justify your excipients, etc., at approval. It doesn't preclude you working with a supplier of an existing formulation to tweak it or optimize it for your use. And where you do use novel excipients, I've given you some indication of how you can work out if they're novel or not, then you might need separate toxicity studies to support the safety. And that is it. Thank you very much for listening.